Don't start yet. Okay. Okay. All right. Good. All right. Sounds good. Okay. Say it again. Is that right? Okay. Okay. Got it. All right. Good morning. Thank you for coming. My name is Sid Bhupati. I'm Chief Operating Officer at Absorption Systems. Today I'll be talking about clarity on transporters navigating the 2012 FDA Draft Guidance. Before I begin, I want to make a quick confession. Here goes. The confession is, I'm a transporter geek. And I work at a company where there are many people just like me, and maybe a few of you in this audience. By no means am I suggesting that I know everything that there is to be known about transporters. All I'm saying is that there is a passion, there is a focus to learn and contribute to this vibrant and dynamic field. So speaking of passions, we added another passion about 7 months ago. And this passion came with the birth of a first child. His name is Vihan. So anytime you have multiple passions, it's always a struggle to balance between the two because they're both equally important, but they are disparate interests. So one way you could approach this is, and this occurred to me, what if I somehow merge these interests? What if I taught my baby boy to be interested in the science that I do for a living? Easier said than done. And that's when I start thinking maybe a better way to approach this if I somehow intersperse my science with illustrations from the books that he likes, fairy tales, cartoons, and so on, to this effect. Not actually a happy outcome. But this exercise was not completely futile. In fact, it was not a wasted effort. I actually learned a few things along the way. And one of the things that I learned along the way was I had this opportunity to reread a lot of books that I had read growing up when I was much younger. And one of the classics that I reread very recently was Through the Looking Glass. This is a sequel to Alice in Wonderland, a book written by Lewis Carroll. And the book is loosely based on a chess game. The book is about a journey into a new dimension where dogged persistence is necessary to emerge on the other side. And along the way, you meet many, many fascinating characters, some likable, some not as much, some inspiring, some impressive, and some who are out there only to create obstacles. And as you traverse, you begin to realize that you're not merely a spectator, that you have the ability to participate in this journey to alter its endpoint. Transporters is very similar. If you have the right persistence, if you have the right preparation, and if you consistently execute, you have the ability to successfully navigate and improve your outcomes. So over the next 30 minutes or so, we will discuss issues that are necessary for improving these outcomes and also maybe meet some characters from Through the Looking Glass that might enhance our knowledge of what is to be expected. So begin by the clinical significance of transporters. As many of you recognize, these matter because they have the ability to alter your pharmacokinetics and your pharmacodynamics. Pharmacokinetics because they alter the extraction ratio of your absorption or clearance organ. What that does, it changes the exposure of your drug substance. When you change the exposure, you could either have decreased efficacy or increased toxicity. Altered pharmacodynamics because transporters are a determinant of tissue distribution, and that can change the efficacy of your drug substance. In addition to that, they can also alter endogenous drug substances such as bile acids, and this can result in cholestasis or idiosyncratic adverse drug events. So by 2006, PGP had become a big deal. And I liken this to the first character that Alice meets as she steps through the looking glass. So it had become fearsome. This character's name is Jabberwocky. But as Alice begins to recognize the character, she realizes that it is not as fearsome as first thought. PGP is similar in the sense. Now when you think about PGP circa 2006 versus circa 2012, you begin to realize that maybe perhaps a bit misunderstood, that many of those drug-drug interactions manifested by PGP are in concert with other drug metabolizing enzymes. It's not PGP alone. And then maybe also the recognition that this can be rendered harmless. Its impact can be mitigated. You can actually build favorable drug-specific properties that can help you circumnavigate around these drug-drug interactions. So why was PGP a big deal back in 2006? The first reason was PGP is ubiquitous. So as in it is your barrier to drug absorption. It can be a determinant of tissue distribution. It can modulate both metabolism and elimination. And also by 2006, there was an increased frequency in tool availability, which led to more identification of drug substrates and inhibitors. All of that combined with the clinical DDI manifested, like your digoxin, quinidine, fexofenadine, ketoconazole, colchicine with clarithromycin, led to a few withdrawals and also a few reapprovals. By reapprovals, these were drug substances that had always been in the market, like colchicine from maybe 500 years ago, but a reapproval is necessary as fatalities were being recognized, which was not due to overdoses of colchicine, but maybe mediated due to PGP or other drug-drug interactions, which also led to increased PMCs and PMRs, requirements for post-marketing commitments, and so on, because of the importance of this transporter. So as with anything that becomes more prominent, there was a regulatory response to this. The FDA put out its draft guidance in the year 2006, in which it said PGP was a big deal. They acknowledged it. And they also said that testing for PGP now had to become much more necessary than it was before. The draft guidance also acknowledges that the previous paradigm of testing was to perform these clinical drug-drug interactions, and these drug-drug interactions could be numerous, they could be expensive, they could be onerous. So they opened up this new dimension of maybe using in vitro tools. So this was the evolving outlook where I compared two panels. On the left panel is a more traditional approach, and this is for a drug substance called aliskiren, which is an antihypertensive agent. So even to investigate aliskiren as a substrate, you had to perform numerous clinical studies, inhibitors of CYP3A4, inhibitors of PGP, combination therapy, co-med strategies, since this was associated with metabolic syndrome, and so on. And maybe as many studies investigating aliskiren as an inhibitor of these different drug metabolizing enzymes or transporters. The alternative was, could we just use in vitro tools as a gate, as a screen, wherein these tools are recognized to be conservative, and in many cases negative findings from such tools can eliminate the need for further downstream clinical investigations. So this is what has happened since the guidance came out about 6 years ago, the first draft guidance. Clearly an increased prevalence of testing. Testing went up from 25% to 42% as you compare two periods in time, 2003 to 2006, and then 2007 to 2010, for new drug applications. Again, since 2006, as in vitro became accepted, there is now an increased frequency in tools used for this. KCO2 makes about 57%, MDCK 21%, and other cells about 22% of all testing that is performed. In addition to that, in vitro PGP inhibition alone, which was about 2% of all tests previously, that increased to about 21%. And that is a really good sign because now there is no downstream clinical study. So one of two things happen. Either your in vitro test indicated that there is no interaction, so that means you have reliable classification, or you have created the possibility for a clinical exception. So in vitro only with no downstream clinical studies. And finally, in vivo digoxin DDI studies only. Between 2003 and 2006, there were 15 in vivo studies performed without any prior in vitro data, because in vitro tools were not as readily available. And now in the new time period, there are very, very few direct clinical studies with digoxin because of the ready availability of these tools. So that's also a very good development, as there's always a screen or gate before you consider a clinical drug-drug interaction study. But as with anything else, challenges do remain. One challenge is how exactly do you set up the right in vitro experiment, because there are numerous considerations along the way. These considerations could include your test systems, these could include the choice of reagents, these could include the substrate and the inhibitor combination that you choose. And in addition to that, since in vitro is this gate, this tool, this screen, the question is, what about translatability as you go from an in vitro study to a clinical drug-like interaction study to a label and finally to the stakeholders, which is a combination of the prescriber and the patient. So there have been significant steps that have been made along the way, but there is much more that needs to be done. Clearly, considerations have matured because there are multiple ways you could approach this. You have the cell systems, you have vesicles, you have overexpressed, but then there is still a question about criteria. Criteria such as your I's and I2s and so on are what are known as the basic criteria, which give you only qualitative information, but there is now the need to have criteria that provides you quantitative information. Can it somehow give you a sense of the extent of impact that can be observed in a clinical study? Labeling has to become more standardized, where the options have to be much more clearly detailed, as in dose adjustment, exercise caution, avoid use, and also this migration from general instructions, as in do not use any PGP inhibitors or CYP3A4 inhibitor, to something that is more specific to the potency of these inhibitors. So much work still needs to be done as we strive towards improved translatability. So while all of this is happening with PGP, there is, of course, much more research being conducted with other transporters, and here are some of those seminal publications, landmarks along the way between 2006 and 2012. So FDA Draft Guidance, there was the ITC Consortium, there's the EMA Draft Guidance, the new FDA decision trees, and so on. And along this path, they have identified other transporters that are now becoming as prominent as PGP. So transporters of increasing prominence can be found at three major sites. The top, of course, is your intestinal enterocyte. To the left is your proximal tubule cell, and also the hepatocyte. The recognition also that transporter kinetics is not a simple enzyme interaction. A simple enzyme interaction is that of maybe, let's say, a drug substance and a PGP, where you can only attenuate absorption or change tissue distribution. But the recognition that transporters actually is a vectorial process. It's a two-step process where there is a basolateral uptake component combined with the apical secretion component. So what that did, as other transporters began to be identified to be prominent, there's also these clinical drug-drug interactions that became manifested with hepatic transporters. I have here a couple of examples with OATP1B1, 1B3, where there is maybe a 6 to 7-fold increase in the victim exposure, and similar idea with renal transporters, OCT2, OAT1, and OAT3. But the story does not really end there. So as you migrate from simple enzyme kinetics to the vectorial process, which is basolateral uptake combined with apical secretion, what you also begin to recognize is that your body's defense against xenobiotics is actually a multi-enzymatic process. So not only do you have these efflux transporters that reduce the load into the cells, but once the xenobiotic makes it into the cells, you of course are hit by both phase 1 and phase 2 metabolism that serve to detoxify. And then these metabolites are secreted again by similar transporters, and this, in a sense, completes your detoxification process. An example of this, repaglinide. Repaglinide is an anti-diabetic agent, Prandon, 2003. And this is what the knowledge was back in 2003, that you should not consider taking itraconazole, which is a CYP3A4 inhibitor, with repaglinide because of the possibility of drug-drug interaction. So there's no questions about potency, and this is as far as the knowledge was back in the day. And then the recognition that repaglinide is not only metabolized with CYP3A4, it is also metabolized with CYP2C8. Gemfibrozil is an inhibitor of 2C8. Now, gemfibrozil matters because repaglinide and gemfibrozil are taken in patients who suffer from metabolic syndrome. And then the recognition that when you somehow, the possibility that if you're on both of these co-meds, repaglinide plus gemfibrozil plus itraconazole, then you actually get an interaction that is not additive but multiplicative, and more on this in a second. And this is where we get into the realm of complex drug-drug interactions. So in addition to 2C8, 3A4, repaglinide is also taken up into the liver by OATP1B1. Gemfibrozil and its metabolite, the gluc, are also inhibitors of 1B1, and therefore that interaction continues to get even nastier. So what we now know is an update in labeling. In addition to CYP3A4, the label also calls for avoiding potent inhibitors of CYP2C8, and it also discusses 1B1 because the panel on the left shows a comparative PK study in 4 different functional haplotypes of 1B1. And as you can see, the top 2, the 521cc and the 521tc, these have reduced activity of 1B1, and therefore there's increased exposure that can result in myopathy and so on. So complex drug-drug interactions can occur when you have concurrent inhibition of enzyme and transporter. And we just saw that with repaglinide and gemfibrozil, 2C8 combined with 1B1. It can occur when you have increased inhibition of drug elimination by use of inhibitors of more than one enzyme. 3A4 and 2C8, you're knocking off both pathways that are responsible for drug metabolism. And it can also occur due to metabolite-mediated drug-drug interactions, as in it's not just gemfibrozil, it is also gemfibrozil metabolite that is responsible for that drug-drug interaction. So what we did learn between 2006 and 2012 is that these 7 transporters, 1 plus 6, have also emerged in a sense. It's not just PGP, but there are other transporters that are equally prominent. And that complex DDI is real, and that is something which might need to be explored as the next frontier as these tools continue to mature. So much like always, the agency's response was a new guidance, and this came out in March 2012. And if Alice were to be looking at this is probably what she would have said. It seems very pretty, but it's so hard to understand. And our job is to maybe elucidate some of that can be made easier over the next 15 minutes or so. So we'll combine this into 3 sections. We'll delineate this 3 sections: substrate assessment, inhibition assessment, and also talk a little bit about induction potential. Substrate potential has to be evaluated for PGP and BCRP because of its ubiquity. Substrate potential has to be evaluated for hepatic transporters if it makes up for more than 25% of total clearance. Same idea for renal. If renal clearance is more than 25% of total, again, substrate potential has to be evaluated. So I begin with PGP and BCRP. You have to have the right test system. And in that test system, if your net flux ratio is greater than 2, and if that ratio is inhibited by 1 or more PGP inhibitors, then you probably have a PGP substrate. And then you start thinking about, is an in vivo drug-drug interaction study necessary and warranted? But here are some considerations to make sure that you get the right outcome. The first is test compound characteristics. And within that, I have 2 categories: non-specific binding and intrinsic permeability. Non-specific binding can easily result in a false positive if you do not pay attention to your recovery or your test compound accumulation and so on. The before and after is the A to B permeability of a drug substance. Before was where we did not correct for non-specific binding. After was where we actually corrected for it. So before, maybe the number was about 2 or 3, efflux ratio was a number greater than 5, clearly appeared like there was a PGP interaction. But after, upon using pre-incubation, upon using 1% BSA, that number increases almost by 5-fold. The efflux ratio disappears. The compounds A to B also beats an internal standard like minoxidil, which now builds in a favorable drug-specific property, which can result in the possibility of clinical exclusions or exceptions. Intrinsic permeability: if your compound has very, very high intrinsic permeability through some preliminary experiment, and this could be through FISChem properties, this could be through a PAMPA evaluation and so on, then sometimes KCO2 may not be the right choice of test systems. And that is what we see on the right panel, where KCO2, A to B is about 10, B to A is about 12 or 14. You essentially get no efflux ratio. So the dots in there indicate that there is no change with or without challenge of an inhibitor, whereas the same compound in MDCK transfected MDR1 has a very, very robust ratio, maybe about 60 or so, which drops to around 5 in the presence of challenge with a PGP inhibitor. So that's another consideration, that your choice of test system may depend upon the intrinsic permeability. Test system choices is the second consideration. Our recommendation is always to consider using the relevant organ phenotype, because that should result in improved translatability as you go from in vitro to in vivo. Example for fexofenadine, in MDCK transfected MDR1, there is just no data for fexofenadine. And that happens because fexofenadine is a more complex drug molecule. It requires this basolateral uptake before being effluxed by PGP. MDCK is not a phenotype of the intestinal enterocyte and therefore lacks those basolateral uptake transporters. Whereas in KCO2, which is now a relevant phenotype, you have both the basolateral uptake assist followed by apical secretion. So KCO2, you get a ratio of about 4, and in PGP knockdown, that diminishes to closer to 1, indicating that fexofenadine is a PGP substrate. And that truly translates because fexofenadine is actually a clinical PGP probe substrate to study drug-drug interactions. Another example is cladribine. This is again to contrast between the relevant organ phenotype versus a single overexpressed test system. In a single overexpressed test system, you actually get a false positive because in MDCK transfected MDR1, the ratio is a number slightly greater than 2. But since 2 is the cutoff, that warrants further investigation. Whereas in KCO2 and P1, the relative efflux ratio is less than 2, indicating that there is not that PGP interaction. The individual efflux ratios in each of these cell lines, KCO2 or CPT-P1, is about 20, also telling us that even though it is not a PGP substrate, there are clearly residual interactions that need to be accounted for, and that was identified to be BCRP when inhibited with KO143, which is a BCRP inhibitor. So the outcome on the right panel, PGP not as relevant, BCRP more relevant, and using a relevant organ phenotype type of test system resulted in identification of that residual interaction. Another element to consider is design. Many times when you're choosing reagents may be non-specific, reagents may be promiscuous, or reagents may have different potencies depending on the probe substrate that you choose. When verapamil was optimized against digoxin, its IC50 is a number between 5 and 10 micromolar. So rosuvastatin is a compound, KCO2B2A is about 12. When run in PGP knockdown cells, that number decreases to about 4, indicating that clearly there is a PGP interaction. But when the same compound is challenged with 100 micromolar of verapamil, which is maybe 10x its IC50 using digoxin as a probe substrate, we do not really see the same type of reduction. Verapamil begins to look more like CPT-P1 or PGP knockdown at a concentration around 500 micromolar. So if you continue to use preset concentrations of inhibitors, which are optimized against a different probe, there is still a possibility of a false negative, because if the only concentration tested is 100 micromolar, your conclusion, erroneous, would have been that maybe this is not a PGP interaction and it could be due to another efflux transporter. The possibility of waiver exists as you build in favorable drug-specific properties. Total efflux, net efflux, is a combination of carrier-mediated, membrane, paracellular, and transcytosis. And in certain situations where your membrane flux is very high compared to that of a co-dosed internal standard, that becomes the dominant parameter and is no longer the rate-limiting step. And in those situations, even when there is carrier-mediated transport, you may not have to worry about its clinical significance. And in absorption systems, we had the ability to do this using minoxidil as the probe substrate, which has a P apparent of about 4 compared to some of the other ones that are used in different places that have much higher permeability values. So to summarize this section, there are numerous considerations. The first thing that you have to think about is, why exactly am I performing this experiment? Is this for a discovery need or for development? What are the properties of the test compound? What is its intrinsic permeability? Should I be thinking about the relevant organ phenotype? Is that relevant organ phenotype even available? What about the specificity of the reagents that I plan on using? And how do I set up an experiment where clinical exclusions are possible? And as you bucket all these considerations, then you start thinking about the actions that are required for these various considerations. Choice of test system, how do you improve recovery so that you minimize these false positives, the right type of calculations so that you compute your effective permeability without bias in the numbers, and also the right internal standards so that you can start building in the exclusions for clinical studies. So moving on to 1B1 and 1B3 substrate. So again, 2 is the operator number. You're comparing your compound's uptake in transfected cells versus vector control cells. And should that ratio be greater than 2, then you have to consider an in vivo drug-drug interaction study. And this is where you get into comparative PK, where you're performing these studies in subjects with various genotypes, various functional haplotypes to get a sense of the importance of this pathway. Same idea for the renal transporters, transfected versus non-transfected. You're looking for a ratio greater than 2. And if that ratio is greater than 2, performing a study in the presence of perpetrators of these different renal transporters. So again, going back to considerations, there could be a tendency to maybe come back and say that one size fits all. And can we use just one set of conditions to identify test compounds that might interact with hepatic and renal transporters? And not necessarily a good idea because of this. Here's an example for known substrates of OCT2. We're looking at 6 different substrates. A couple of them score really well when you use the threshold of 2. Other ones are borderline, and that would include pendolol, propranolol, and maybe amantadine. Why that happens, why one size does not fit all, is for a couple of reasons. Non-specific binding, which is the panel on the left. So if you only use the lowest concentration and the shortest incubation duration, it is possible that even in your non-transfected, you lose so much to the apparatus, it might appear like it is being taken up inside the cells. And that's why there is no distinction at the early time points. But if you now allow the experiment to go for a longer duration, you start seeing separation between the transfected and the non-transfected. Another possibility could just be the robustness of transfection. Maybe you just don't have a great test system to begin with, and you need a much longer duration for separation again between the transfected and the vector control. And sometimes it comes down to transporter properties. When you're thinking about renal transporters and the OATs, compounds that are substrates of these transporters are organic anions, the small molecules that are hydrophilic, and they do not accumulate easily. And as you can see in the example on the right, for the OAT substrates, as you start going towards longer durations, the 10 and 20 minutes, you start scoring better, the difference between the transfected and the non-transfected. And the clone 1 and the clone 2, the difference is the robustness of transfection. And that's why clone 2 performs better, and also with increased experimental duration. So going back to that pendolol experiment, so if you now come back and actually set up a matrix with different concentrations, with different incubation durations, you start seeing a more robust ratio. 1 micromole at 20 minutes gives you a number which is greater than 3, not the borderline where sometimes it may score, sometimes it may not. So here is my remark on test system considerations. I go back to Alice for a second. The White Knight is one of Alice's buddy during her journey, and he meets her at stage 7, which is just one stage before Alice is ready to be crowned a queen. So as I said, a well-wisher, well-meaning, but limited mental faculties, as in he thinks of himself as an inventor, but probably not the best inventor that is out there. So he has this box on the horse, and he looks at Alice admiring this box, and he proudly proclaims that this is to keep my clothes and sandwiches. And as you can see, I carry it upside down so the rain does not get in. And Alice exclaims that, did you know the lid's open? So the idea behind this is he's so concerned about protecting it from the rain, you're not necessarily looking at other considerations. So even as you optimize, as you're trying to provide meaningful results, you cannot focus on just one important property, but you have to take a more well-rounded type of approach to obtain meaningful results. So moving on to inhibition potential. From the ITC white paper, inhibition was to be assessed for PGP and BCRP because of the ubiquity. Inhibition was also to be assessed for 1B1 and 1B3 because statins were so ubiquitous and there's a possibility of that interaction. But they had come back and said that for renal transporters, inhibition potential may depend on your clinical tenet, may depend on your co-met strategy, no longer. FDA Draft Guidance from March 2012 has come back and said that inhibition potential has to be assessed for every one of these major transporters. And the reason that has changed is because many of those drugs highlighted there, metformin, the protease inhibitors, methotrexate, these are all important drugs taken by large sets of population, and therefore it is necessary to also assess for inhibition potential of these renal transporters. So moving on now to PGP and BCRP inhibition. Differences from before is I is no longer based on unbound concentration. I1 is based on the total Cmax. And I2 is your highest dose strength divided in 250, which is different from the guidance in 2006. So as you perform inhibition assessment, you determine the IC50 value, and you have certain thresholds, a cutoff for this IC50 value, which result in you taking or not taking the next step, which is the clinical drug-drug interaction study. So there has been an identity crisis with this, because I has certainly evolved over the years. What started off as total plasma Cmax is now in different places. For a short duration, there was an emphasis on unbound plasma Cmax. For a short duration, there was emphasis on using the highest clinical dose. And now there is also a lot of conversation about using the concentration in the enterocyte, because maybe that is more representative than the concentration in the lumen because of solubility issues and so on. So even though there is this identity crisis and I continues to evolve, what is important here is that I does matter, because that is how you set up your basic criteria, and this example will illustrate why I matters. From a recent FDA publication, this is 11 compounds for which in vitro and in vivo data exists, as in your clinical drug-drug interaction. Anytime you perform a clinical drug interaction with digoxin, what you're looking for is AUC-I, AUC in the presence of inhibitor, over AUC in the absence of inhibitor being a number greater than 1.25. So when your AUC or Cmax is greater than 1.25, then yes, you have a clinical drug-drug interaction. So this is how your numbers look like. Using unbound concentration, there appears to be a lot of false negatives, and false negatives is the last place that you'd want to be. Using total, 2 false negatives. Using I2, which is the highest dose strength in 250 ml, 2 false positives, and then of course the right 2 columns are your clinical data. To summarize that graph, if only I1 was used, there is no way that is working, because your accuracy rate is only 55% and you get 5 false negatives. 5 false negatives as in your in vitro tool is not picking up an in vivo clinical drug-drug interaction. Using total or combined, accuracy improves dramatically, but you still get 2 false negatives, and that is a safety concern. When you use I2, no false negatives, you get 2 false positives. FDA may not care as much about this, but as pharmaceutical scientists, we also want to reduce the incidence of false positives, because that results in unnecessary testing. The reason false positives take place, one is solubility, because this is based on highest dose strength divided by 250 ml. You might never reach that type of solubility in the lumen. We talked about this before. There's an example for a compound called etraverin, where its IC50 in vitro was determined using Taxol as a probe substrate. But in vivo, you're using digoxin. So of course, there could be difference in numbers. Sometimes your compounds could be weak inducers. And also, the interaction could be so borderline that depending on how many times you run the study, you might get a slightly different outcome. And that's an example of Marav-Rag. So those are just some of the things to appreciate. So if you use a combination of I1 and I2, you'd still be left with 2 false positives. IC50 determination has received a lot of attention. And then there was this working group formed in 2008. And their objective was, can we somehow standardize this so that we can start building better cutoff criteria, better and improved translatability. So they looked at various considerations. This was a consortium of almost 24 companies that came together to run these different IC50s for 16 compounds, using Keiko2 cells, using overexpressed, using vesicles, and so on. So some of the considerations was, can we identify the best test system? Can we identify the best calculation technique? How do we minimize inter and intra-lab variability? And is there some way we can internally calibrate using your data to enhance your predictability? Their findings, there is no best test system or equation. Every one of these has variability. KCO2 and vesicles tend to produce lower IC50 values. It's not necessarily a bad thing. It just means you're conservative. But again, from a development perspective, you also want to avoid unnecessary in vivo testing. And there are all these tools, statistic-based tools called principal component analysis, AUC prime plots, accuracy plots to define your cutoff values, what's known as receiver operating characteristic analysis. All of this can be done to enhance accuracy, and I'll talk about that in the next slide. So from the FDA guidance, their criteria, I1 over IC50 is greater than 0.1, I2 over IC50 is greater than 10, and we just saw that accuracy was 8 out of 10, right? From the 11 compounds, 1 compound has been removed, ranolazine, because this is something that the IC50 working group had also worked on. So what the IC50 working group did, they had 16 interactions that were assessed. They formulated the rules and applied it to the 11 compounds that the FDA had put in the literature. And when they applied their rules, they came up with a different criteria, I1 over IC50 greater than 0.03, I2 over IC50 greater than 45, just using the principal component analysis, AUC prime, ROC, and so on. And now their accuracy, they start doing a little better, 9 out of 10, and certain labs actually did 10 out of 10. So what can be done essentially is that since there is no best test system, there is no best equation, what is of paramount importance is that internal calibration. At this point, there are maybe 33 compounds for which in vitro and in vivo data is available or can be made available, and it's imperative that you go back and validate your test system based on this information, come up with your own criteria to enhance your overall accuracy rate and reduce your false positives and have 0 false negatives. Another important consideration is solubility, because as your eye continues to evolve and become higher and higher, it is imperative that you have the right test compound concentration and solution for a meaningful assay. One way to do that is by adding excipients. The first time we approached excipients, we approached it maybe in the wrong manner. We came back and said that it is important that none of our test system criteria changes, that we need to have the same integrity values, we need to have the same rank order, we need to have the same PGP functionality, and so on. So we ended up using excipients that were in the range of 0.002%, which essentially was counterproductive, because what we're hoping for was to take advantage of the solubilization of these excipients. So upon further consideration, just going back to the statement that I had made about the white knight, you cannot really optimize one parameter at a time. What we recognized was that you had to optimize a combination of both solubility, impact, and sensitivity. So even if our PGP substrates, like a digoxin that you would use to assess inhibition, even if its ratio diminished from a 20 to a 10, we would still consider that to be acceptable, adequate sensitivity for assessment of PGP inhibition. What is more important was to make sure that you could actually bump up the concentration of excipients to attain meaningful solubility. So in this example, the excipient concentration goes up about 250-fold, 0.05% PS80 actually can be used to dramatically increase solubility of many, many test compounds. And also, as can be seen here, the inhibition potency of CSA does not really change with or without excipient, even though you're working with less sensitivity than in an unaltered test system. The IC50 values are comparable, 1.5 versus 2 micromolar. So moving on to 1B1 and 1B3 inhibition, the biggest differences here are, again, total Cmax versus unbound Cmax, and the R value, which is your correlation in vitro to in vivo, is now 1.25 instead of 2, which is different from the ITC white paper. The justification for these changes, the FDA provided this recently. They went back and looked at 28 combinations of substrates and inhibitors to ask the question, which one is a better criteria, the ITC recommendations or what the FDA is thinking about today. So the left panel is the ITC white paper. unbound over IC50 greater than 0.1. So 28 combinations, and they got 5 false negatives. Again, right off the bat, it's a bad sign because the last thing you want is false negatives. Of those positives, which would be the true positives and the false positives, anything that had a number greater than 0.1, they came back and applied the R criteria. So when they applied the R criteria of 2, of this 21 compounds, 6 more became false negatives. So essentially, you had 11 false negatives, not very good, from the combination of these 28 substitution inhibitors. When they went back and applied their I total to first the 28, they got 0 false negatives, which is exactly where you want to be. There are only 2 false positives, not a very high number. And then when they applied their R value on those 26, the 24 plus 2, they actually got 5 false negatives. So at this point, the FDA's Transporter Scientific Interest Group, which is responsible for setting up these criteria, is even beginning to question, do I need the R value? Because applying that resulted in 5 false negatives, whereas just using I total or IC50 gave you 0 false negatives. For renal transporters, similar idea. This is now based on unbound Cmax. So you determine the IC50 using your transfected cells. And then if the ratio is greater than 0.1, additional investigation using a sensitive probe substrate. So some considerations for IC50. It is important that this is performed under linear probe substrate conditions. If you do not perform under linear probe substrate conditions, you get Km values that are higher than what need to be. If you get Km values that are higher than they need to be, then you get IC50 values that migrate in the wrong direction, and that results in the possibility of false positives. I finally want to end with one slide on induction. Not much has changed between 2006 and 2012. Transporters can be induced. Now there is a possibility that transporters other than PGP can be induced, but there still remains this lack of in vitro tools for determination of induction potential. So to give an example of a tree, if you were to assess induction of PGP, which occurs through the PXR, CYP3A4 is used as a surrogate or as the gate. So in the beginning, you would assess in vitro CYP3A4 induction. If that is a yes, CYP3A4 induction in vivo, and only if that is a yes, CYP3A4 induction in vivo, would you consider any type of PGP induction in a clinical setting. So improving translatability still remains the biggest challenge, and this is in a sense what is to be expected in the coming years as you go from in vitro all the way to the prescriber and patient. So these are all areas where dramatic improvements have been made. We have gone from just applying basic criteria to much more mechanistic approaches, such as using the R value, using PK/PD, using fraction transporter, and so on, which can, in a sense, quantify the extent of interaction that can be expected in the clinical study. Our recommendations have also become much more specific. It is no longer just avoid use, but they could be exercise caution, they could be dose adjustments, and there's also many more nuances that have become tools for the patient and the prescriber as you're trying to assess, well, what do I do if I had to take gemfibrozil with meropeglonide and so on? It's not one broad bucket of just do not use these co-meds. Strides need to be made in these areas. We spoke about test system calibration. Applying the FDA's universal cutoff may not necessarily work for your internal test system. Complex drug-drug interaction, because most of the current understanding is towards simple or vectorial, but how do you build an in vitro tool that can actually assess the net impact of an enzyme and a transporter? How do you extrapolate some of these learnings to unstudied drugs? Does every drug have to be studied clinically, or is there some way you can use PBPK to make those types of extrapolations? And finally, how does the FDA become more consistent when asking for these PMCs and PMRs? What data is that based on? And also overall stakeholder education so they can appreciate how these can be manifested. So in my closing thoughts, I want to just leave you with a mindset that might be required for continued and sustainable success. This is a conversation between Alice and the White Queen, where Alice talks about not being able to believe in impossible things, and the White Queen comes back and says that why sometimes I've believed in as many as 6 impossible things before breakfast. I think most of us lie somewhere in between, but this is just something to think about. That's all I had to say. Thank you. I know that you're looking at some media placements and so on. So I was like, let me just take this opportunity. I've not presented this to a few internally yet, so I thought I'd just do that. This was, I presented this in Jersey. Oh, the right. New Jersey program campus in this country. Right. Yeah. I stopped it.